go then. What I'm going to be talking about today is the effect of melatonin on heart rate. And my thanks to the organizing committee to invite me for this, and happy to be a keynote speaker. I'm not sure what that entails, but I hope I live up to it. What I'm going to be talking about is the effect of melatonin on heartbeat in the fly. And let's see if we can advance here. There we go. Might be a little clunky, I think. Using the Drosophila as a model, and I always have to do this because when people start talking about cardiology, they don't think about flies. But flies have been a useful model. Realize there have been 10 Nobel laureates given to people working with flies, including T.H. Morgan, who discovered genes and chromosomes. Getting right down to business, I also pointed out that a lot of cardiac work has been done, and what's of interest to us here is that 75% of the genes that have been determined to be at the root of disease in humans have analogs in flies. So to business. Take a look at the heart. I'll just go through this briefly to show you what I've been working with. I work with the pupil heart. This is a pupil right here. The Drosophila heart is a simple tube. It has inlets, at least in the pupil, 6 in the pupae, more in the adult, which pick up hemolymph. And importantly for us here, hemolymph does not contain hemoglobin. It's not really true blood. This is a little bit different, of course, from working with the vertebrate heart in that it supplies oxygen to the body, whereas flies use a tracheal system. Importantly, though, it means you can have really, really badly mutated hearts and have the flies still live. And that's made life a lot easier for me in terms of working with the fly and figuring things out. The system is myogenic. Hearts and insects were thought to be neurogenic for many years because they were working with a very poor model to show that it was myogenic. It is very clearly myogenic. Here's a scanning EM showing the heart. There's the heart running down through a dissected pupa. These are the heart cells down through. These are pericardial cells, which are functionally neurocytes in the insect. Here's a very close-up of a transmission showing you the heart. Here's a hemocele of the fly and the hemolymph inside the heart. It's a single layer thick, and it's an interesting heart. It has a pacemaker at the most posterior end, and we'll see it has a lot of very close analogs to the vertebrate. Now, another benefit, of course, is, as I say, really sick flies don't die. Let's take a look at what I did with it. I've been recording with from pupae. This is a very early stage pupa, right after the third larva transitions. This is what it takes, a half a metric ton of equipment to record something from something the size of a period in a sentence. But it works very well for us, and we have quite a lot of really fine data from it, which you're going to see some of. This is a typical recording. This is a minute of optically recorded data from a fly. Now, optically is important. These flies live. Now, when you do an experiment with flies, you have a bunch of animals that you're not sure after you've done a cross what you've got until they become adults. And if you have to kill them to record their hearts, you got nothing to work with. You don't know what you had as a genotype. This means in this system, you still have the genotype, and that animal lives and can be bred. So for those of you who have worked with genetics and understand fly genetics, that's pretty essential to what we're doing. Now, as to analyses, I've done it. I have to show you. Most of you, if you're familiar with cardiology or familiar with EKGs, know what they look like. I had one on my title page. This is the first analysis I do, other than looking at the raw data, which you just saw. This is an autocorrelation. You take the datum, the time series, and do a correlation with itself. And then you lag it by one time frame. And if it's rhythmic, these sequential peaks occur. It's a very powerful analysis, which is not done with EKGs. I should point out that my current research, I'm also a professor of mathematics, is to make it possible to do autocorrelation analysis with EKGs. And I'm collaborating with cardiologists on that right now. But I myself am the individual who came up with this as a rhythmicity index, the height of the third peak, counting the leg 0 as 1. That height of the third peak is the rhythmicity index, and it gives you a good idea of how regular the heartbeat is. Now, the other thing I pioneered was the use of this particular analysis. It's far superior to Fourier analysis for picking up spectra. This is a spectrum of the heartbeat you saw initially and represented in the autocorrelation. This is a spectrum showing that the frequency of the heartbeat is around 2 hertz in this particular animal. And notice a very clean spectrum. I'll show you this is a wild type animal. Fly hearts are not always that rhythmic. And this is an example of probably as arrhythmic and a wild type as you're going to see. And just to show you, this is the autocorrelation. The rhythmicity index here is only 0.221. And so again, another piece of information showing that the autocorrelation is an extremely powerful way of judging rhythmicity in and output of the fly heart. Now, another way of looking at it, which is common in cardiology, is to look at the interbeat interval. So if you see this irregular heartbeat up above, and below it is the interbeat interval, just the literal time between the peaks in the analysis. And you can see how well it corresponds to the irregular portions up here. So this is going to be an important analysis as we move forward. What I did as background is my lab spent many years looking, using genetics and pharmacology to work out the basics of the cardiac pacemaker in the fly. And here's a series of mutations showing you what you can get. This one's the one we're going to look at more carefully. This is a calcium-gated potassium channel. That's wild type. That's a mutant slowpoke fly. Shaker and Ethiogogo are potassium channels. But using these analyses and using these techniques, we came up with a model. I just wanted to show you this briefly. This is a corellogram of a wild type fly, and this is a corellogram of a slowpoke fly. That's what his heart looks like. It barely beats, but as I said, they do not have to worry about oxygen transport. So the flies live, called slowpoke, because they meander very slowly. They're not very healthy, but they do live. You can breed them, and you can study their hearts. And here's a comparison of the spectra from a wild type fly in the solid line and a slowpoke fly in the dashed line. To show you the effectiveness of this is, don't worry about the details, but as I say, I'm working in the mathematics department. A colleague there and I worked up a series of differential equations which modeled the behavior of the basic families of ion channels which comprise the fly pacemaker. You have a leaking channel, okay, a potassium leak channel, okay, which triggers a calcium spike. And then you have a calcium-gated potassium channel, which repolarizes. And here's where slowpoke it comes in, because it's repolarizing the plasma membrane of the pacemaker. Now, what about melatonin? That's what we're all about here. And I had an undergraduate wander in to do wanted to do an honors project, and she said, you know, you found in your lab the serotonin speeds up the heart. Maybe melatonin slows it down. Wouldn't that be interesting? Well, doesn't slow it down. But let's look at melatonin effects in general. I apologize. It's the only list I'm going to give you. Hypnotic sedative effects. It has powerful antioxidant activity. It's anti-aging in rats. But critically for cardiology, it protects against ischemia reperfusion injury from reactive oxygen species, because it's a very, very effective antioxidant. And endocrine system regulation, reduces blood pressure. And what else does it do? Well, here's a little bit of what we did. We have used the concentrated micromolar, 46 nanoliters injected 200 to 1 dilution by the hemolymph. So it's not very much. It's not very far off what's used in treating reperfusion injuries in human hearts after infarct. Record for 10 minutes, inject, and then reposition, re-record for another 10 minutes. These are interbeat intervals. This is wild type. Notice it's not all that regular, okay? And we'll talk about that in a couple of minutes. And after a few minutes, look what happens. And this, you don't need to do tables or statistics or anything. That's what melatonin does to rhythmicity of the fly heart. And as you'll see, we'll talk about in a few minutes, like throwing a switch. Okay, now I'll show you why we picked 30-second intervals after you see what the data looked like, and you'll see how I justified doing what we did. This is raw data from one 30-second interval through the first portion. And here's what the heartbeat looks like. This is raw data. This is what you get from your machine that you're using to record it. You know, it's self-explanatory. You could almost use this to time a wristwatch. Autocorrelation before, this is the same fly, the same heart, before and after melatonin, and not a lot of melatonin. I mean, that's, I have to say it myself, it's remarkable. These are the spectra, before and after, and you saw the difference between a regular and irregular spectrum. And here's the interbeat interval from those regions amplified, showing the difference. The same fly, the same heart, before and after melatonin injection. Now, of course, we did the stats. We, also professor statistics. We took every 30-second interval before and every 30-second interval. So that's how we chose the intervals to look at under greater scrutiny. Now, we also did a dose response, and this is where we came up with 1,000 micromolar concentration to use. That's where we get a peak response. So we did that work beforehand, but I wanted you to see that after we did the, you saw what the results looked like. So this is just to justify what we did. Now, what does it do? We asked the following questions. Maybe it's just speeding up the heart rate and it appears more regular. But melatonin does not affect the heart rate at all. Serotonin does, and it does not affect regularity. Maybe what we're doing is we're titrating out reactive oxygen species. No, ascorbic acid, which does the same thing, has no effect on regularity. So it's something else. We hypothesize that it's receptor mediated. Melatonin receptor agonist luzendol eliminates the effect. So if you shut down the receptor, and we were the ones who discovered the melatonin receptor in the fly using this technique, eliminates the effect. If you shut down the receptor using RNAi, you can shut down the transcription of the, or the effective transcription of the gene. The orphan receptor of that receptor eliminates the effect. And here's this slide again. This does not look at all like the effect of the buildup of the concentration of the substance. No, it looks like somebody threw a switch right there. And that is almost a signature of a receptor-mediated effect. Now, back to this slide. What happens if you give melatonin to a fly like slowpoke that has a really nasty heartbeat? The same thing, exactly the same thing. This is a fairly good slowpoke fly compared to what we have, and this is what it looks like. It's as more regular than normal as wild type. So melatonin is literally rescuing the defect. It's rescuing a cardiac defect, producing a regular heartbeat. Now, let me talk about effects. Supposing you're going down the Autobahn at some ridiculous velocity and you exceed the capacity of your Audi's engine and it blows. Now, is there any way you're going to get that motor to take you down the road? No, you're not. It's not going to happen. It's completely shot. I'm not sure that that's an Audi motor, but it might be. It doesn't matter. It's almost a metaphor. The only thing that's going to work is a new engine. So, supposing the fly has another pacemaker, And what we're doing is that we're switching, using melatonin, switching from the pacemaker in the that the fly normally has on the plasma membrane to something else. Well, that opens a can of worms. There is excellent evidence that there is a second pacemaker in cardiac cells, including in mammals. And it involves two main ion channels, or anodine receptors, which release calcium from the sarcoplasmic, endoplasmic calcium, endoplasmic reticulum, and returned by calcium ATPase, returns it to that the sarcoplasmic reticulum. And this is a little bit more complicated than that. But we have a mutation in the fly that I've worked with, and I was one of the first people to publish on it. It's a temperature-sensitive mutation in the gene which encodes this calcium ATPase, and it affects heartbeat very severely after a heat shock. So we hypothesize that's what's happening is that when you give melatonin to the fly, and what it's doing is it's changing the communication between the plasma membrane oscillator, which we've delineated, and you saw the evidence of that, and the internal cytosolic oscillator, maybe even turning it over to that oscillator to control heartbeat. Now, what is the evidence for that? Well, this is evidence that has not yet been published, which I'm working on. Take a look at this right here. This is what circa looks like, and this is the rhythmicity index, as you saw from the correlation, autocorrelation of the fly for the 10 minutes after heat shock. So the heartbeat is very badly destroyed when you mess with the sarcoplasmic reticulum oscillator, with the cytosolic oscillator. Importantly, if you also give a melatonin injection, it doesn't help. So unlike all of the other mutations we tried, and slowpoke wasn't the only one, but unlike all the other ones that we tried, there was no effect. It did not restore heartbeat in that particular mutant. So there's further evidence for a hypothesis that's what's happening is either changing the communication between the two oscillators or switching over to that as a spare oscillator. Now, another can of worms, which speaks to this. This is normal looking heartbeat, but heartbeat isn't necessarily perfectly regular in healthy individuals. This is heartbeat in beats per minute, and you can see this is a little bit like our beat per minute. This is normal heartbeat, but in congestive heart failure, it becomes very much more regular. And a number of people have hypothesized that the heartbeat, it has inherent chaos in it. This is, again, a can of worms. It has not been resolved necessarily, but is the heartbeat chaotic? Okay. Well, chaos, here's a chaotic oscillator. It's a pendulum with a main structure here and a second structure here. And it's chaotic because you cannot predict what it's going to do into the future. And this is, can this be true with the heart? Well, we ask the following question. If this is chaos, how can you change the chaotic oscillator into this non-chaotic oscillator? So I contend that our melatonin results are a way of helping to answer the question of whether there's chaos in heartbeat. And finally, my invitation to cardiologists is to explore this. There is some very scant evidence that melatonin use in helping with infarcts actually increases the rhythmicity of the heart. So one of the things we found, one of the things you normally do as a pharmacologist is to look for analogs. 2-iota melatonin, which is an analog of melatonin itself, is twice as effective in changing the regularity of the fly heart rate. So we hope that potentially some cardiologist out there is going to take the bait and begin working with mammalian models and find some potentially a way of using melatonin or some of its analogs to increase the regular heart rate in human cardiology patients. So thank you, and thank you from the mountains of Maine, where it's still very cold. If there are questions, you can, I know we're a little bit behind. Why don't you try to look at keep my eye on the chat, and if anybody has any questions, I'll look for them there. Thank you very much.